Minutes to take your questions. Again, there is a microphone on the left side, and please address your questions regarding the subject to our panel. And we'll probably take 15 minutes before we go for lunch, and we have a lecture also scheduled with lunch, and some of us will also be available after lunch to take some more of your questions.
I want to thank Dr. Barcellos-Hoff for giving a wonderful talk. This question is directed to you. Current drug therapies like tamoxifen and Herceptin are obviously targeting differentiated cells, whether it be ER positive or ER negative, and they've shown a lot of success to it. But of course, one of the problems is that they are not obviously killing the cancer stem cells. So could these drugs actually be inducing differentiation in these cancer stem cells into something more aggressive that could be resistant to these drugs and thus causing recurrence of the cancer later on?
Well, that's an interesting idea. Whether tamoxifen and HER2 targeted therapeutics are actually changing the composition of the tumor. I think tamoxifen has some interesting effects as being an anti-estrogen in perturbing the balance between cell types. But whether it actually pushes cells in one lineages or changes the stem cell compartment, I think is an open question until we have better markers for stem cells in these tumors. So right now, all the evidence is very indirect, and of course, those specimens are difficult to obtain. So I think we're going to have to wait maybe a couple more years to answer that question. It's a good one, though.
Yes, I have a question about adult bone marrow stem cells. And I know you can increase the level in the blood by using some drugs. Is there a potential for a therapeutic benefit if you could do that and increase the levels in the blood, that it would then maybe migrate to those areas in the body that are needed for maybe they can differentiate? I think that's at issue, whether these adult stem cells could differentiate and be of therapeutic benefit.
Is this on?
Yeah.
Okay. So there has been quite a number of studies over the last 5 to 10 years on the potential of bone marrow cells to cure a number of different diseases. And there's two types of diseases. There's diseases, for example, in the brain that you need the secretion of a certain product, okay? So in that case, you might be able to use other cell types, basically jam in some other cell types that don't integrate, don't actually form any of the structures in the organ to provide some of the factors that are needed. There's limited potential for that, but it is at least theoretically possible. There was a series of studies suggesting that bone marrow cells could make everything from neurons to cardiac cells to even eggs and sperm. So you don't even need eggs and sperm. You can actually just get them from bone marrow. Most of those studies, in fact, all of those studies, have systematically been disproved. Now, that doesn't mean that bone marrow cells won't have some usage if we do something fundamentally different. But the way the science stands right now, just taking the cells and expecting them to do something that their program does not really, their programmed language does not have in it, is really not a realistic way to cure any disorders. So there's been a number of clinical trials, the largest of which have been on using bone marrow cells from a patient to improve outcomes after heart attacks. There are now large trials that have discounted that effect. So as it stands, just replacing neurons with bone marrow cells isn't going to work. Bone marrow cells are pretty restricted. They've been known, their potential has been known now for 30 or 40 years. There are some limited applications if you need some factors secreted. But in general, any sort of degenerative disease outside of the blood system, they wouldn't be valuable for.
Okay, thank you.
I found Dr. Hoeffler's discussion of his use of adult stem cells in skin cells to be very, very helpful. And I was thinking of you know, the various sources of adult stem cells also being in the human placenta, you know, as well as the embryonic cord and amniotic fluid. And also there has not been very much discussion on all of the successes in human disease, treating human disease with the adult stem cells. And I think that that is something really remarkable to pursue. I think that adult stem cells there should be much more research with adult stem cells. And there was a law that was passed in California to promote this by one of our Democratic legislators. The other thing is that my understanding is that one of the problems with human embryonic stem cell research is the tendency to form tumors or cancer. And I think that maybe adult stem cell would have, you know, an advantage over human embryonic stem cells in that regard. So these are some of the questions that I have to raise. Thank you.
Who wants to take it?
Yes, I agree with that, and I mean, I certainly see a lot of potential for the adult stem cells, but at the same time, the embryonic stem cells are also very appealing. You know, if you, as a scientist growing these up in dishes in the laboratory, what we see is that the embryonic stem cells are more stable and they'll go through more population doublings than the adult stem cells. Adult stem cells are difficult to isolate, and they lose their stemness, we say. You know, after being passage from one culture dish to another, it's easy to lose their stem cell abilities. So there are really reasons to remain committed to the embryonic stem cell work, but I agree that there's a lot to learn about adult stem cells, and I think they'll be very useful as well.
One thing I'd like to add is that adult stem cell research has never been excluded from federal funding. There's always been a lot of funding for adult stem cell research, so that I think there needs to be more, and especially as I think as big as there was a crisis in embryonic stem cell funding, which CIRM has largely addressed, there is an overall crisis in federal funding for science that is unbelievable right now. Right now, we're looking at success in getting federal grants for those who have MDs or PhDs that are hovering continually around 8%. When I started as an assistant professor 10 years ago, it was around 22 to 25% of applications were funded. We have reduced our funding to a level that is just unsupportable, and there is a tremendous need to fund medical research.
So I just want to add a comment about your question about the teratoma or tumor using different type of stem cells. So of course, adult stem cells, that's one you would assume have less risk. But then again, you should remember when people using embryonic stem cells for therapy, first you already differentiate these cells into lineage committed or already differentiate cells. And if you graft those cells into the body, if you can eliminate all the embryonic stem cells, then you should as safe as adult stem cells.
My question is for Dr. Zeng. Is there any evidence that Parkinson's disease is inherited, since I have several members of my family affected?
Yes, there are some genes, for example, like Parkin, that's one of the genes that are actually being found, and also other genes called alpha-synuclein and other genes that actually has been found. If people have defect in these genes, they have a slightly higher risk for Parkinson's disease. But Parkinson's disease is really more sporadic. It's not really like a typical hereditary genetic disease.
Thank you.
Hi. And that brings me to my question of determinants for disease. So it sounds like that the success of cellular therapies will depend on our ability to understand the genetic determinants of diseases in the cases of like Parkinson's, where we know there's some genetic factors involved. So is there an equilibrium in terms of the rate of science and understanding the genetics of disease to match up with how fast stem cell research is progressing? Is there some sort of link between the two to try to figure out how, so for example, in the case of someone who does have a disease because they're genetically predisposed, if we're to use a cellular therapy that will basically cure them of their disease, do we necessarily need to alter their genome or just put in a copy of the normal gene? And if we do that, will we successfully restore an organ's function, or will we always have sort of a level of disease process existing?
That's an interesting question. So actually, you are talking about two therapies. Now, we talk about cell therapy and gene therapy. For those diseases that has a genetic defect, even you do with an somatic cell transfer or you use induced pluripotent cells, let's assume that can be usable one day in the long future or near future. So you still need to correct those genetic defects. That means you should be able to do either a very targeting change, like I've talked about the gene targeting. You want to correct exactly that disease copy and then replace with yours. That actually means you're going to go to two steps of therapy. First, you go cell therapy and then gene therapy. Then that would be a long shot. For the non-genetic defect, the disease, okay, you can use your own cells. And that with the induced pluripotent cells or somatic cell nuclear transfer, that may solve the issue of immunoinjection. So that's two therapies.
Okay, and then just one other question. So for Dr. Pera, do we know in the maternal cytoplasm what is causing the reprogramming of the genomes?
No, not really. And in fact, the induced pluripotent, the systems that are being used right now are kind of bypassing potentially what the egg has always done. The fact that there's people in the room is a testament to the fact that reprogramming happens. It happens naturally. But there are a number of efforts and a number of labs looking at the cytoplasmic determinants. So far, people have been focused mostly on what's in the nucleus that is probably talked to by the cytoplasm.
Dr. Pera, my question has to do with, it's pretty far-fetched, but the intersex kind of person who has no gonads. Is there any possibility that there might be some way to put them in after the fact?
So to put them in would be a little bit tough. But there are, so if you look at whether or not you could make germ cells, eggs and sperm, which is really what the organ system, the gonadal system is supposed to do, I believe that is not too far away. And the idea is not so much necessarily to be able to reproduce in a dish as to be able to understand the generation of birth defects, the generation of problems in the germ cells. many, many things are unique to humans. So the generation of Down syndrome, if you look at a simple bred yeast, it will make what's called a meiotic error that ends up with an extra chromosome, about 1 in 10,000 divisions. In humans, by the time a woman would reach the age of about 40, that's on the order of 1 in 10. It's just an incredibly different system of regulating things. And so understanding the molecular basis for those things is really important. It's important for diagnosing, and it's important for just correcting things.
Thank you very much.
Mm-hmm.
This question is directed to Dr. Pera again, though I guess anyone can jump in. Since everyone in this room pretty much knows the potential of stem cell research in the medical field, I'm wondering about how long would it take for the U.S. to overcome the political and the ethical dilemmas that affect our understanding of human development at the embryonic level?
So Louise Brown, you're too young to know probably, but Louise Brown was born in 1978, so a lot of people in the room perhaps might remember the birth of the first in vitro fertilization baby. The situation at that time was not that birth was not heralded universally as something that was desirable. Shortly after that, there was something called the DUC Amendment, which went into was passed by Congress and went into any federal funding. So it regulated that federal funding was explicitly prohibited for embryo research. Okay, what you had now was a situation where IVF was legal, but embryo research was illegal. Okay, so now you allowed the practice of a certain type of medicine, but you mandated it that it be done without knowledge. And that's one of the most incredible things to do, because at that point, the culture methods, many of the ways to actually grow embryos became stagnant. And we know that this process is not optimized. Women go to get IVF, and it is often, if a woman is older, especially 2, 3, 4, 5 embryos will be placed into the uterus. Okay, that is done solely because we don't know what a good one looks like. So let's put a bunch in. Okay, now what happens if all 5 of them take? Occasionally you see something on TV where somebody had quintuplets. That's so rare. Mostly those are miscarried, or in order to save the life of the woman, you have to have an abortion. So we have a system that doesn't serve the right to life community very well. It doesn't serve women very well, and it continues to allow IVF, which is something that people who want children, and 10 to 15% of couples cannot have children. Okay, so we're looking at something that's not rare. But we're mandating that no embryo research be done. Now, what was interesting is that in 2001, nearly 20 years later, President Bush said, and I'm extending this basically to embryonic stem cell research. That's the addition that he really did. What surprised me about that, I mean, I really, as the director of the Human Embryonic Stem Cell Research Program, I care very much that we said that we allowed it to be said that we won't do human embryonic stem cell research. And what happened was there was a rise, and people said, no, that's not okay. What I regret is that we didn't say, no, that's not okay back in 1978, when we said that women can continue to have embryos of subpar quality put in as kind of a test for whether or not they're good and have miscarriages, but that's okay. So I think that we have a system that has been in place for a long time. Now, in very optimistic ways, I think that human embryonic stem cell research affects a lot of people, and you can see the potential value. And I hope that as we move forward, people will begin to see the value of understanding early embryo development. And I'm not a fan of extraordinarily widespread embryo development experiments, but just that we regulate them and we're responsible. And so we will see if it will change, but it's been since the late 70s, early 80s, and so it will take a concerted effort to change that. And I think it will take a concerted effort on those who care about the general women's health.
Thank you.
Our last question.
Thank you all for the presentation today. My question is very much related to what you just answered for that lady, but it hasn't been answered. I can't figure out how, you can say you can't legislate morality, but I heard CIRM say that you're covering ethics towards this. Does the FDA have anything to say? Why should a bunch of politicians try to legislate science and morality? And does the FDA do anything? From what I understand, they're being cut back continually. And what will be the answer? Will it be the medical profession, the research profession, or the pharmaceutical profession running our future in this country?
Well, I think that what's been interesting, what's been interesting is that nothing was made illegal. It was funding that was cut off. That's tricky. That's trickier. I think that people uprise against saying that this is illegal, you can't do that. And in fact, Congress was never able to make stem cell research illegal, but they were able to take the funding away. And that's a really clear distinction. Now, I think my colleagues can answer this better, but there's definitely a place for, I don't know if it's large pharmaceutical, but we definitely need private companies in this area. It is extraordinarily difficult, even at Stanford University, whose endowment is front page news. It's extraordinarily difficult to fund an FDA clinical trial.
All right, so congratulations, you have survived the first part of the morning session. I want to thank our panel, and as a reward for you, you'll be able to join us for lunch. And as another reward, you can have a lecture on the ethics of stem cells as a bonus. So please head outside, make sure you have your ticket, make sure you fill the evaluation form. There will be students escorting you to the cafeteria, and we'll see you there. Thank you so much.